Welcome to the deep dive. Okay, so today we're tackling a really core topic, how sex gets determined genetically speaking.
Right.
We're taking the whole chapter on sex determination and sex chromosomes from Klug's concepts of genetics and basically boiling it down for you.
Yeah, the goal is to give you that college-level overview, step by step. We'll go from those first sort of fuzzy observations of chromosomes right through to the actual molecules making it happen.
And it's all about genetics controlling differentiation, right?
Absolutely. We often default to thinking X and Y chromosomes, the heteromorphic ones. But this dive shows it's really about specific genes. Sometimes they're on sex chromosomes, sure, but sometimes they're elsewhere, or even responding to things like temperature.
It sounds like there's a lot of variation.
Oh, incredible variation. And it evolves surprisingly fast. So we'll trace how our understanding developed.
Okay, let's jump into that history. Where did the chromosome link first pop up?
Well,
It's pretty early, wasn't it?
yeah, remarkably early. Back in 1891, Hermann Henking noticed this weird structure in some insect sperm. He didn't know what it was, just called it the X body.
Just X, like the unknown variable.
Exactly. Then Clarence McClung saw something similar in grasshoppers, called it a heterochromosome. But it wasn't until 1906 that Edmund B. Wilson really connected the dots.
And Wilson nailed down the basic systems.
He did. He described two key patterns. First, the proteinor mode, like in some butterflies. It's XX for females, but males are XO. They just have one X.
Right, so they're missing a chromosome entirely.
A sex chromosome, yeah. So the male makes two kinds of sperm. Half get the X, half get, well, nothing, no sex chromosome. If an X-less sperm fertilizes an egg, you get a male XO offspring.
Huh. Okay, so the male gamete dictates the sex there. What was the second mode?
That's the ligaeus mode, named after the milkweed bug. This is the XXY system we're more familiar with, like in humans. Females are XX, males are XY. The male produces X-bearing and Y-bearing sperm in, roughly equal numbers.
Right, that sounds more standard. Now, you mentioned the male gamete dictating sex. Is there specific terminology for that?
Yes, and it's important. The sex that produces different types of gametes regarding sex chromosomes, like the XO or XY males in these examples, is called the heterogametic sex.
And the one producing uniform gametes.
That's the homogametic sex, like the XX females in those systems who only make X-bearing eggs.
But it's not always the male who's heterogametic, is it? I feel like I remember something about birds.
You're absolutely right. It's flipped in birds, moths, some fish, and reptiles. There, the female is heterogametic. To avoid confusion, we use a different notation, ZZZWW.
So ZZ males and ZW females.
Exactly. Males are the homogametic sex, ZZ, producing only Z-bearing sperm. Females are heterogametic, ZW, producing Z-bearing and W-bearing eggs.
Okay, that clarifies the different chromosomal setups. Let's bring it to humans. We knew the diploid number was 46 by 1956, XX female, XY male. But what was the big question then?
The core question was, does the Y chromosome actively make someone male, or is maleness simply the result of not having a second X chromosome? It wasn't obvious.
And how do we figure that out?
The answer came from studying individuals with unusual sex chromosome numbers, usually resulting from errors in meiosis, something called non-disjunction.
Ah, where the chromosomes don't separate properly during egg or sperm formation.
Precisely. And two syndromes were particularly revealing. First, Klinefelter syndrome. These individuals have 47 chromosomes. They're XXY.
XXY. So they have female chromosomes and a male one.
Kind of. They have two X chromosomes plus a Y. Phenotypically, they are male. They have more genitalia, but often underdeveloped tests and some feminized features like breast development, which is called gynecomastia.
So the Y chromosome overrides the two X's.
That's the crucial point. Even with two X chromosomes, the presence of a single Y is enough to direct development towards maleness. It showed the Y carries some kind of dominant male determining signal.
Okay, so what about the opposite situation? No Y chromosome at all.
That leads us to Turner syndrome. These individuals have just 45 chromosomes. They have a single X, and that's it. 45X.
And they develop as?
Female. They have female external genitalia, but typically underdeveloped ovaries, short stature, maybe some other physical signs like webbing on the neck. So no Y means female development, even with only one X.
So Klinefelter and Turner together really sealed the deal.
Absolutely. The Y chromosome determines maleness in humans, period.
There are other variations, right? Like XXX or XYY.
Yes, briefly, triple X, 47,000 XX, results in females who are often perfectly normal, though sometimes taller or with some learning difficulties. And 47,000 XYY males are generally taller than average. There were some early flawed studies linking XYY to aggression, but it's now clear most lead completely normal lives.
So the why is the key. How does it actually work at the molecular level? When does this decision happen?
It starts surprisingly early, around the 5th week of embryonic development. At this stage, the gonads are actually bipotential. They haven't committed yet.
Meaning they could become either tests or ovaries?
Exactly. They have an inner part, the medulla, that can form testes, and an outer part, the cortex, that can form ovaries. It's waiting for a signal.
And that signal comes from the Y chromosome.
Precisely. It comes from a single gene on the Y called SRY, which stands for sex-determining region Y.
The master switch.
That's the perfect term for it. SRY encodes a protein called TDF, the testis-determining factor, and TDF is a transcription factor.
Meaning it controls other genes.
Right. It binds to DNA and turns other genes on or off. Around week 7 in an XY embryo, TDF gets produced and kickstarts testes development, likely by activating genes like SOX9.
So SRY starts a cascade.
A whole cascade. Once the testes start forming, they begin to produce hormones. One crucial one is Müllerian inhibiting substance, or MIS.
And I guess that inhibits the Müllerian ducts.
You got it. In all early embryos, you have two sets of ducts. Müllerian ducts, which can form the female reproductive tract, uterus, fallopian tubes, and Wolffian ducts, which can form the male tract, epididymis, vas deferens. MIS, produced by the new testes, causes the Müllerian ducts to degenerate. Without MIS, they'd develop. At the same time, testosterone produced by the testes supports the development of the Wolffian ducts. So SRY just lights the fuse.
It's amazing one gene can do that. What about the Y chromosome itself? It's pretty small, right?
Tiny compared to the X. The Y has maybe 75 genes, while the X has somewhere between 900 and 1,400. Structurally, the Y has these small regions at the tips called pseudoautosomal regions, or PARs.
Pseudoautosomal, meaning kind of like non-sex chromosomes.
Sort of. These PARs contain genes also found on the X chromosome, and critically, they allow the X and Y to pair up and even recombine or swap bits of DNA during meiosis. That pairing is essential for them to separate correctly into sperm cells.
And MS of the Y.
The other 95% is the MSY, the male-specific region of the Y. This is where SRY is located, along with other genes mostly involved in sperm production and male fertility.
I remember reading it was once considered a genetic junkyard.
Yeah, a functional wasteland was the term. But work by David Page and others showed it's actually quite complex. It has these weird palindromic sequences DNA that reads the same forwards and backwards.
Like madam?
Exactly, but with DNA bases. It turns out the Y chromosome uses these palindromes to repair itself since it doesn't have a fully homologous partner like other chromosomes do for most of its length.
It can't use the other copy for repairs.
Right. So it seems to fold back on itself and use these palindromic regions to fix mutations, especially in the genes vital for fertility. It's a really clever mechanism.
And the MSY is evolving fast.
Very fast. The human MSY structure is surprisingly different from even our closest relative, the chimpanzee. And recent research links the Y not just to sex and fertility, but also things like paternal age effects, mutations increasing with father's age, and even suggests loss of the Y chromosome in some cells, like blood cells in older men or smokers, might increase risks for certain cancers. It's definitely not junk.
Okay, shifting gears a bit. We've established sex determination, but there's a consequence. Females have 2 X chromosomes, males have 1. How does the cell handle that genetic dosage difference?
Great question. If females expressed all the genes on both X chromosomes, they'd produce twice the amount of X-linked proteins as males. For many genes, that imbalance would be lethal.
So there has to be a fix.
There is, and it's called dosage compensation. The first clue came from Murray, Barr, and Ewart Bertram in the late 1940s. They noticed a dense, dark spot in the nucleus of cells from female cats, but not male cats.
The Barr body.
Exactly. The Barr body, or sex chromatin body. It turns out this is a highly condensed, inactivated X chromosome.
Inactivated, so it's just shut down.
Largely, yes. The rule that emerged is the N1 rule. In any given cell, all X chromosomes except one are inactivated and become Barr bodies.
So a normal XX female has 2 - 1 Barr body.
Correct. A Turner female, 45X, has 1 - 1, 0 Barr bodies. A Klinefelter male, 47,000 XXY, still only keeps 1 X active, so he has 2 - 1 and none Barr body. An XXX female would have 2 Barr bodies.
That's neat. How does this inactivation happen?
That brings us to the Lyon hypothesis. proposed by Mary Lyon in the early 60s. She pieced together several key ideas. First, the inactivation happens randomly in each embryonic cell early on around the blastocyst stage.
Randomly, so either the mother's X or the father's X can be shut down.
Yes, it's a coin flip in each cell lineage. Second, once that choice is made in a cell, all its descendants maintain the same inactive X. It's fixed for that cell line.
Which means the female body is a mix.
Exactly. It leads to mosaicism. Adult females are literally mosaics of cells where the paternal X is active and cells where the maternal X is active.
And the classic example is
Calico or tortoiseshell cats. The gene for orange versus black fur color is on the X chromosome. If a female cat inherits one X with the orange allele and one with the black allele,
he's heterozygote.
right, then, due to random X inactivation, she'll have patches of cells expressing the orange allele where the black X is off, and patches expressing the black allele where the orange X is off. That's what creates the mottled coat pattern. Males, having only one X, are typically just one color, black or orange.
That makes perfect sense.
Yeah.
It's a visible outcome of this random inactivation. How does this cell actually do the silencing? What's the mechanism?
It's complex, but it centers on a specific region on the X chromosome called the X inactivation center, or SIC. Within the SIC is a crucial gene called XIS, X inactive specific transcript.
Is that a protein?
No, and that's what's really cool. XIST produces a long non-coding RNA, an lncRNA. It doesn't get translated into protein. Instead, this RNA molecule literally coats the X chromosome that's going to be inactivated.
It paints the chromosome.
In a way, yes. It spreads along the chromosome and recruits proteins that chemically modify the chromatin, condensing it down into that inactive Barr body state, effectively silencing most of the genes through blocking transcription.
Wow. And how does the cell know to only inactivate one X? Or all but one?
That counting mechanism is still being fully worked out, but it seems to involve the GIS regions somehow communicating, maybe briefly pairing up. Other genes within the GIS, like 6, which is spelled backwards and acts antagonistically, and ZEIT, are also involved in ensuring only one X chromosome remains active. It's a very precise regulatory system.
Okay, that covers mammals pretty well. But sex determination isn't always about the Y, as we mentioned. Let's talk about fruit flies, Drosophila.
Right, Drosophila is a fascinating contrast. They also have XX females and XY males, superficially like humans, but here the Y chromosome has nothing to do with determining sex.
Nothing? So what's its function?
It's essential for male fertility, making functional sperm. But an XO fly, one with just a single X and no Y, develops as a male. It's sterile, but it's physically male. And an XXY fly is a fertile female.
So if the Y isn't the signal, what is?
It's the genic balance theory, worked out largely by Calvin Bridges. What matters is the ratio of X chromosomes to the number of sets of autosomes. We call this the XA ratio.
XA ratio, how does that work?
A normal female is XX and has 2 sets of autosomes, 2A. So her XA ratio is 2x.2A equals 1.0. A normal male is XY, or just X, and has 2 sets of autosomes. So his ratio is 1x.2A equals 0.5. It's this ratio that determines the sex.
So 1.0 means female, 0.5 means male. What about other ratios?
They can happen, especially in experimental settings. Ratios between 0.5 and 1.0 typically result in intersexes, individuals with mixed characteristics. Ratios above 1.0 might be metafemales, often weak, and below 0.5 metamales.
That's a completely different logic than the Y-dominant system. How does the fly actually measure this ratio?
It's ingenious, really. The primary signal seems to be the dose of certain genes located on the X chromosome itself. The master switch gene responding to this ratio is called sex-lethal, SXL.
Sex-lethal, okay.
Sex-L is activated only when the XA ratio is 1.0, i.e. in females. If the ratio is 0.5 males, XSL stays off.
And what does sex-XL do once it's activated?
Sexis is a splicing regulator. It controls how the RNA transcripts of downstream genes are processed. Specifically, it affects the transformer TRA gene and the double-sex DSX gene. This whole process uses alternative splicing.
Meaning the same initial gene transcript can be cut and pasted in different ways?
Exactly. In females, active sex-XL ensures that the TRA transcript is spliced correctly to make a functional TRA protein. TRA, in turn, directs the splicing of the DSX transcript to produce a female-specific DSX protein. This female DSX protein then regulates downstream genes to implement female development.
And in males?
In males, XXL is off. So the TRA transcript is spliced into a non-functional form, no TRA protein is made. Without TRA, the DSX transcript gets spliced in a default different way, producing a male-specific DSX protein. And this male DSX protein directs male development. It's an elegant cascade controlled by RNA processing.
Very clever. Is this XA ratio system seen elsewhere?
Yes, a similar principle, though simpler, operates in the nematode worm C. elegans. They don't even have a Y chromosome, they have two sexes. Hermaphrodites, which are XX, and males, which are XO. Again, it's the XA ratio, 1.0 for hermaphrodites, zero point 0.5 for males, that determines the outcome.
Okay, so we've seen Y dominance and we've seen genetic balance. What's the third major category?
The third one takes us away from chromosomes as the primary determinant altogether. It's temperature-dependent sex determination, or TSD.
Found in.
Many reptiles, all crocodiles, most turtles, quite a few lizards. Here, the incubation temperature of the eggs during a specific critical window of development determines whether the embryo becomes male or female.
Wow, so the environment is directly controlling sex. Are there patterns?
Yes, there are three main patterns described. Case 1, low temperatures produce females, high temperatures produce males. Case 2 is the opposite. Low temps yield males, high temps yield females. And case 3 is more complex. Extreme temperatures, both low and high, produce females, while intermediate temperatures produce males.
And the temperature difference can be quite small.
Often incredibly narrow, sometimes just a few degrees Celsius can completely flip the sex ratio of the offspring.
How does temperature actually influence gonadal development? What's the molecular link?
It seems to involve temperature effects on enzymes involved in steroid hormone production. A key player is the enzyme aromatase.
Aromatase, what does that do?
Aromatase converts androgens, like testosterone, typically thought of as male hormones, into estrogens, female hormones. In many TSD species, higher aromatase activity is correlated with ovarian development. Temperature seems to influence the expression or activity of aromatase or related genes in the developing gonad.
So temperature tweaks the hormonal balance.
That appears to be the mechanism, yes. Pushing the bipotential gonad down one path or the other by altering the local steroid environment. It can even get really complex, like in the Australian bearded dragon lizard Pogona viticeps.
What happens there?
They actually have a ZZZW genetic system, like birds. Normally, ZZ is male, ZW is female. But if you incubate the eggs at very high temperatures, it can override the chromosomes. Genetically male, ZZ embryos, can develop into perfectly functional females.
So they have a genetic system and a temperature override.
You'd like it. It shows just how flexible and, frankly, weird sex determination can be across the animal kingdom.
Okay, that's a lot to cover. Let's try to recap the main themes from the chapter. We looked at how sex is determined, starting with the basic chromosome systems like XXXO and XXXY, and the ZZZW reversal.
Then we focused on humans, where the Y chromosome, specifically the SRY gene, is the dominant trigger for male development, proven by syndromes like Klinefelter XXY and Turner 45X.
We discussed dosage compensation in mammals, the whole Barr body and Lyon hypothesis, where one X is randomly inactivated in females to balance gene expression using that cool X-sized T non-coding RNA.
Right. And then we contrasted the Y-dominant system with the genic balance system in Drosophila, where the XA ratio is key, interpreted through that elegant alternative splicing cascade involving sex-L, TRE, and D-sex.
And finally, we touched on environmental control with temperature-dependent sex determination in reptiles, likely mediated by enzymes like aromatase.
So 3 very different strategies: a master switch gene on a sex chromosome, a ratio of chromosomes, or an environmental cue.
It really highlights the diversity. Do you have a final thought for our listeners, something to chew on based on all this?
Well, what strikes me is how fast these mechanisms seem to evolve. Think about the human Y chromosome. Its structure is quite different from the chimps, despite us being closely related. And you see completely different systems pop up in different lineages. Birds use ZW, flies use ratios, turtles use temperature. It makes you wonder, if something as fundamental as sex determination can change so readily, what does that say about the stability and evolution of other core genetic processes? It suggests maybe the how of sex determination isn't as constrained as we might think, allowing evolution to experiment quite a bit.
That's a great point. It's a reminder that even fundamental biology is dynamic. Thank you for guiding us through that chapter. It's a complex topic, but hopefully this deep dive made it clearer.
My pleasure.
And thanks to all of you for listening.